Hello, my name is Anushka Bharwani, and I'm currently a junior majoring in Pharmaceutical Chemistry and minoring in Mathematics. I currently conduct research in the Hartigan O'Connor Lab.
The HOC lab is in the Department of Medical Microbiology and Immunology. One of the major projects is vaccine development, which I will discuss more in this video. As we know, the SARS-CoV-2 virus infected millions and led to a worldwide pandemic. Fortunately, effective vaccines have been developed and authorized for emergency use. The vaccines authorized for use in the United States target the spike protein of the virus. The spike protein is a target because it plays a role in viral attachment, fusion, and entry. The HOC lab is developing a SARS-CoV-2 vaccine targeting the receptor binding domain. The RBD is a part of the spike protein of the virus and allows binding to ACE2 receptors. We want to test if the vaccine-induced RBD antibodies are effective in preventing binding onto the ACE2 receptors and thus neutralizing the virus. My role in the project started by cloning the antibodies expressed from the vaccinated animals. Before I get into the cloning, I'll do a brief overview over the structure of an antibody. An antibody is a protein that can bind to antigens in the body, and an antigen is a foreign substance in the body that triggers an immune response. Upon binding, the antibody provides protection through various mechanisms. There are 5 types of heavy chains and 2 types of light chains. The 2 light chains are kappa and lambda, and the heavy chains are IgG, IgA, IgD, IgE, and IgM. We can determine the type of chain based on the constant region. For the purpose of this project, we are solely interested in the IgG antibodies. Some applications of antibody cloning include establishing a cell line producing the antibodies, sequencing and mapping the antibodies, and running various assays. This technique is really important because monoclonal antibodies have many therapeutic uses. In this particular experiment, we are using antibody cloning to further characterize and understand the immune response induced by RBD. Now that we've gone over the basics of an antibody and what antibody cloning is, let's get into how this is actually done. We first run a PCR on cDNA samples in order to amplify the heavy and light chain regions. We then run a gel in order to verify that we did amplify a heavy chain and to determine if we have a kappa or lambda light chain. We then purify the PCR product and quantify the DNA concentration using nanodrop. Next is the actual cloning step. We take our backbone vector and integrate our insert into the vector. From here, we perform a bacterial transformation with the plasmid. The next day, we would pick colonies and run a PCR to verify that the bacterial colonies grown on the plate do indeed have an insert. Then comes a plasmid extraction, and finally, the samples are ready to send out for sequencing.
Thank you for watching, and a huge thank you to Dennis and Nate.